Hello, my name is Fisayo Omonije. I'm a sophomore at Johns Hopkins University, and I've been very fortunate to be one of the inaugural CUSP program interns. And I'm here today to share my experience during my NIH summer internship, where I interned at Oregon Health and Science University and worked on the SenNet project, which is a collaborative effort between Oregon Health and Sciences University researchers and researchers at Pacific Northwest National Laboratory. And kind of what the whole overarching goal is to define senescence characteristics and do single cell proteomics on the proteins. What I've been doing at OHSU has been working on optimizing senescence staining in murine or mouse skin. So just a little bit of background on senescence. Senescent cells are when cells undergo permanent growth arrest. So this matters to us because senescent cells are key determinants of many age-related diseases. The one kind of at the forefront of my internship was cancer, because I was working at the Cancer Institute at OHSU, but there's a whole plethora of others, like type 2 diabetes, osteoarthritis, and the list goes on. So the researchers at OHSU and PNNL decided that a workflow needed to be created so that we can identify these cells and kind of quantify which proteins are in them and figure out what proteins these actually are so that we can learn more about them. Thus was born the SenNet project. And just getting a little bit into the specifics of what we do at each side. At OHSU, the crux of the internship and the crux of what they've been working on has been optimizing many different stains so that we can see these cells in the mice tissue. Just the anti-GFP, P16, and SA beta gal in our mouse model, the fast mice, which I will explain as we go. And then once the staining procedure is fully optimized, OHSU sends those samples to our collaborators at PNNL, where they will be doing single cell and spatial proteomics to kind of figure out what's inside these cells and what proteins are there so that we can, again, learn more about it. And they're doing this by liquid chromatography and mass spectrometry. So just the methods that we used, if you look on the first two rows of this diagram, these are what we kind of did at OHSU. And then the bottom row is the PNNL side, and I'll touch on that. So as I mentioned, the fast mouse model is what we use to enable Cre-mediated senescence. So the fast mouse model differs from a normal mouse in that we feed it tamoxifen, which is a drug that enables the Cre enzyme. Now, if you look into this diagram at the right of the first right before the arrow, you'll see that all these mice have this genome called the LOX/STOP-LOX P27 IRES GFP cassette. And how tamoxifen works in these mice is that it activates the enzyme Cre. And what Cre does, as you can see, it cuts out anything between the two LOX sites. So it cuts out the stop codon so that P27 and GFP can be expressed. P27 is pretty much what causes senescence, and GFP is a protein that we use to be able to see them on the microscope when we do these immunofluorescence and SA beta gal stains. So again, just the methods here, the whole staining process, taking the tissue from the skin on their back, embedding it, freezing in OCT media, cutting the samples on the cryostat at a very small thickness, staining them, and then imaging these samples to where we get these pretty images, as you'll see at the end of the second row. And then again, I'll get into the PNNL side towards the end of my presentation. So this is just an atlas of what we should be seeing in our stains. As you'll see, the middle and the left image are immunofluorescence stains, and the right image is an SA beta gal stain. On the left, you'll see this is our what we call a control, or in many cases, a secondary antibody alone image. And what we want to see here is just very little fluorescence. You can see that there's a lot of blue on the image, and the green, as you'll see, is fluorescence, which is kind of tells us that there are positive senescent cells in these tissues. So again, we should be looking for not much fluorescence on the secondary antibody or control slides. And then in the treated samples, we obviously want to see this fluorescence. And this green lets us know that we do have senescent cells. So this is just an ideal kind of best case scenario, what we look for. And then on the right is an SA beta gal stain. Now this differs from an immunofluorescence stain because the SA beta gal, again, it's an enzyme that helps us see senescent cells. But it's very important that for these experiments, the SA beta gal master mix, which we use on the stain, be kept at a neutral pH, or else we won't be able to see the cells. So again, the arrows kind of point at the positive cells, or the senescent cells in the stroma, which is what we're looking for. So now I'll just get into all the different stains that I did. I'll start with the anti-GFP, which was the first one that I did. As you can see, the secondary and alone, we don't want to see fluorescence. So hey, first glance, this looks great. Yeah, no fluorescence, no GFP expression. So we expect to not see any senescent cells here. And then we did this iteration, we used the Cre positive and Cre negative treated mice. Now these photos are interesting because as you can see, there is green. It's not very fluorescent. So the lack of fluorescence here is that we kind of knew that the staining procedure didn't work as well. And what's also contributing to this is that there's a high level of background. These are just some images, but we took many. And in most of the images, there was a very high background. And high background pretty much just means that we have a lot of, I guess, positivity of GFP in the tissue. And we'd rather see it scattered at a cellular level rather than the entire tissue, either being too bright or too dim. So again, just a lot of kind of almost a dud. I hesitate to say dud, but we'll look at the Cre positive samples. And again, it's kind of the same thing. We do see background, and it's just not as bright as we want it to be. So this told us two things about our stain. One, that the staining procedure in this particular instance wasn't up to scratch, that it wasn't giving us the results we wanted. And two, as you can see, there's no real difference between a Cre negative and a Cre positive sample in the amount of positive cells that we would like to see. So ideally, in the Cre negative and the Cre positive, we expect to see senescent cells in both, but the Cre negative, we expect to have way fewer, just because in the Cre negative, you're going to have senescent cells in the mouse. That's just how aging works. But when we do the Cre positive, because we've induced the senescence, we expect to see far more. And in the anti-GFP, it just wasn't the case. So we took from this that we needed to optimize our staining procedure further, and that we need to kind of look at the way tamoxifen is being administered to the mice, because if we don't have good tamoxifen administration, we don't have that senescence that we would like to see. So that was the anti-GFP stain. And then we moved to the P16 stain. Again, P16, like P27, it's pretty much a senescence marker, and it pretty much causes senescence. And PMA is like GFP. It allows us to see senescent cells in the tissue when we take these samples. So as you'll see here, these are control samples, and you can see that there are senescent cells in the control samples. So again, this is kind of not what we would like to see, but I'll just say a little aside. A thing that I learned at this internship is that science is science. Like, you're not going to get great data all the time, and it's still good to know where you're at so that you can continue to optimize these things. So these were our control samples. Again, not what we see ideally. And then these are our PMA treated or positive samples. Again, you'll see that there's really not much of a difference in the quantity of positive cells we have in the fluorescence. But as you can see here, the staining did work. There's a little background, and we can actually see that there are senescent cells in the tissue once we take the images. So one side of the equation is kind of done, but again, we need to look at how senescence is being seen and the levels that we're getting between the control samples and the positive samples, and what we need to change in our tamoxifen administration. So we redid the P16 stain, but instead of doing the stain on PMA-treated mice, we used it on Cre mice. So mice that have the enzyme that it has been mentioned before. And I'd say this stain was probably the most encouraging stain that I did over the entire summer, because it gave us really good results that we would like to see. So as you'll see in our secondary antibody alone or control sample, there's little to no fluorescence. And in our Cre negative samples, you see that there still is some senescence, but it's nothing compared to the Cre positive. And this is exactly what we want to see. So you'll see on the graph to the right, 4.75, that's the amount of average positive cells per sample that we counted, versus the 2.82 for the Cre negative. So this was great. We presented this data to our collaborators at the PNNL, and this was really encouraging data. So that is it for the P16 staining and the immunofluorescence stains. Next, we move to the SA beta gal stain. And this was kind of the same, I guess, idea. You'll see on the graph here, this is just another demonstration of how the process works to get these images. And I'll just move to the staining images, what we're looking for. We see senescent cells in the tissue. But again, you're looking at a control sample and a PMA-treated sample. And when we counted the cells, we didn't see much of a difference again. So we validated that the stain procedure was working, but again, the tamoxifen administration is not giving us a difference in these senescent cells in the actual tissue. So that was a takeaway from our SA beta gal staining, that we had the procedure working, but same thing. And that leads me into our conclusions that, again, the SA beta gal and P16 staining procedures have been fully optimized, but the anti-GFP, unlike these two, needs continued optimization. We talked to our collaborators at PNNL, and they gave us suggestions on, for the specifically anti-GFP stain, what we could do to optimize the stain, whether that be the minutia in the actual procedure, changing the fixation media, whether the slides are put into 95% ethanol or 10% glycerol. That could help us a lot, because before we were doing paraformaldehyde treatment. So just little things like that could lead to better cellular expression. And then again, the different approaches to tamoxifen administration. During my time at OHSU, in the Rulin lab, it was a tamoxifen chow that we used in our procedure. But I guess post me, they'll be doing an injection or a topical where they just take the hair off the mice and then rub a topical right on their skin so that we can see it when we take the tissue samples. And hopefully these differences lead to more robust identification of these senescent cells and a change in the control versus the actual treated samples. So the future directions for the SenNet project between OHSU and PNNL. At OHSU, to validate both sides of this workflow, we know we have the staining working in most of our iterations, but again, getting that tamoxifen administration. And then at PNNL, microdissecting the single senescent cells in their areas, digesting the protein using the nanopots chip. And what the nanopots chip is it allows you to use many cells at once. Whereas without it, you'd be doing digestion of just one singular cell. The nanopots chip allows you to do over 240. So it really speeds up the process of getting these cells, I guess, in what we need to get them in. And then doing liquid chromatography and mass spec to analyze the proteins that are in the cells and give us kind of a more holistic understanding of what we're working with and how these cells can eventually contribute to cancer and getting therapeutic remedies for these age-related diseases. And outside of the entire SenNet project, just continued collaboration with the whole SenNet Consortium and laboratories from different universities. I know that the OHSU and PNNL folks have been working with Yale University senescence leaders and also people at University of Washington, just to kind of get, again, a more holistic understanding of the characteristics of senescent cells so that we can understand them better. I'd like to take a moment to acknowledge many people. This internship, I'd say, was extremely formative. I want to first thank all the OHSU and PNNL staff, Megan Ruland, who was my PI in the Rulin lab, my mentor, Julia Unsworth, and Liliana Pasatolich, who was the lead researcher at PNNL and continues to be. Without them, I couldn't have done any of this research. So I really give so much credit to them. And then I'd also like to thank the SenNet Consortium, the NIH, and everybody who's been a part of the CUSP program, the students, the people who've been leading us, too many names, Kay Matisse, Linda Manonia, Natasha Soliman, and I know there's so many more I could thank. So again, just thank you guys for this opportunity. It's really been formative in kind of helping my passion grow for the research sciences. And I know I'll take this experience with me as I plan to go on to med school and continue to immerse myself in the biomedical sciences. So again, just big thank you to everyone involved. And it's been a great summer.